bacteria, which means that they can take on different shapes, round like a coccus, or coccobacillary, which means somewhere between a spherical coccus and a rod-like bacillus. The most common species that cause disease in humans are Ehrlichia chafensis, which causes a disease called human monocytic ehrlichiosis, or HME, and Anaplasma phagocytophilum, which causes a disease called human granulocytic anaplasmosis, or HGA. Now, Ehrlichia and Anaplasma have a thin peptidoglycan layer, so they don't retain the crystal violet dye during gram staining. Instead, like any other gram-negative bacteria, they stain pink with safranin dye. Both are non-motile, non-spore-forming, and obligate intracellular, which means that they can survive only inside cells. Finally, Ehrlichia and Anaplasma don't grow on routine culture media, and they need to be cultivated in vitro in different cell lines. So Ehrlichia chafensis can be isolated in DH82K9 histiocytic cell line, and Anaplasma phagocytophilum can be isolated in promyelocytic leukemia HL60 cell line. Now, Ehrlichia and Anaplasma enter circulation following a tick bite, and once inside the body, they infect circulating leukocytes. Ehrlichia primarily targets monocytes and macrophages, and it infects them using tandem repeat proteins, or TRP. These bacterial proteins bind to proteins found on the surface of the cell, and they induce phagocytosis. So basically, they make the cell gobble up the bacteria. Anaplasma, on the other hand, primarily targets neutrophils, and infects them with the help of a p-selectin glycoprotein, which binds on the p-selectin glycoprotein ligand 1, or PSGL1, found on the surface of neutrophils. This activates an intracellular pathway that leads to reorganization of cellular actin, which leads to phagocytosis, allowing Anaplasma to enter the cell. Once inside the cell, both Ehrlichia and Anaplasma live in an early endosome, which normally merge with lysosomes to kill invading bacteria. But in order to merge with lysosomes, this early endosome needs to become a late endosome. So the early endosome contains a protein called RAB5A, which is involved in the recruitment of RAB7A and the maturation of these compartments into late endosomes. However, Ehrlichia and Anaplasma don't let that happen, so it stops the production of RAB5A. How they do that is still unknown, but the end result is that the early endosome cannot become a late endosome, so it does not merge with the lysosome. Thus, bacteria avoid intracellular killing by lysosomal enzymes. Additionally, Anaplasma can avoid intracellular killing by inhibiting the production of reactive oxygen species produced by neutrophils to destroy invading bacteria. Also, it has the ability to destroy XIAP, a protein which normally inhibits neutrophil apoptosis, or programmed cell death. So by destroying XIAP, it stimulates neutrophil death. Now, when they're safe from intracellular killing, Ehrlichia and Anaplasma replicate inside early endosomes by binary fission. This means the bacteria splits into two identical copies. And if it sounds similar to mitosis, well, it is. But the term binary fission is used to describe division of prokaryotic cells, which don't have a nucleus, and therefore some steps in replication are different from mitosis. So they form microcolonies inside endosomes, called moruli, which are mulberry-like inclusions. Finally, when they've replicated enough, they cause cell lysis, leading to release of bacteria into the bloodstream, and from there they can disseminate into multiple organs like the lungs, heart, kidneys, liver, or central nervous system. So both Ehrlichia and Anaplasma can cause serious complications if left untreated, like respiratory failure, heart failure, renal failure, and septic shock. Neurological complications associated with Ehrlichia infections are mainly meningitis and meningoencephalitis. Complications include demyelinating polyneuropathy, when the myelin sheath of neurons in the upper and lower limbs is destroyed, resulting in nerve damage, and brachioplexopathy, which is when there's damage to the brachial plexus, where upper limb nerves originate. Now, Ehrlichia is transmitted by Amblyomma americanum, or the Lone Star tick, which is usually found in the northeastern and midwestern US. On the other hand, Anaplasma is transmitted by Ixodes tick found in deer and white-footed mice. There are three types of Ixodes ticks involved in the transmission of Anaplasma: Ixodes pacificus, found in the western US, Ixodes ricinus, found in Europe, and Ixodes scapularis, found in the eastern US. On a side note, Ixodes scapularis can also transmit Borrelia burgdorferi, which causes Lyme disease, as well as Babesia species, which cause babesiosis, so co-infections are possible. Symptoms of both human monocytic ehrlichiosis and human granulocytic anaplasmosis include fever, chills, severe headache, myalgias, nausea, vomiting, and loss of appetite. There's also an atypical rash that looks like red splotches or pinpoint dots located on the trunk, but it spares the hands and feet. With meningitis and meningoencephalitis, there might be fever, headache, stiff neck, confusion, hallucinations, and seizures. With demyelinating polyneuropathy, there's tingling or numbness in the toes and fingers, weakness of the arms and legs, fatigue, and abnormal sensations. And with brachial plexopathy, there might be numbness, pain, and weakness of the shoulder, arm, or hand. Diagnosis is done with a peripheral blood sample that identifies the characteristic moruli in monocytes and neutrophils using Wright stain. PCR can also be done to identify bacterial DNA. Serological tests, like indirect immunofluorescence assay, or IFA, can be done to detect IgM and IgG antibodies against Anaplasma or Ehrlichia. Routine lab studies can show leukopenia, thrombocytopenia, anemia, hyponatremia, or low concentrations of sodium in the blood, and mildly or moderately elevated hepatic transaminase levels. When there's central nervous system involvement, a cerebrospinal fluid analysis can be done, which can identify the moruli. Finally, treatment is done with antibiotics like doxycycline as the first drug of choice, or chloramphenicol as an alternative. Alright, as a quick recap. Ehrlichia and Anaplasma are tick-borne, gram-negative, pleomorphic bacteria. They're non-motile, non-spore-forming, and obligate intracellular. Ehrlichia infects monocytes and macrophages, leading to a disease called human monocytic ehrlichiosis, or HME. And Anaplasma infects neutrophils, leading to a disease called human granulocytic anaplasmosis, or HGA. Diagnosis is done by identifying in a peripheral blood smear the characteristic moruli found in monocytes and neutrophils using Wright stain, with PCR, which can identify bacterial DNA, and with serological tests like IFA. Treatment is done with doxycycline as the first drug of choice, or chloramphenicol as an alternative.